Hey guys, today we're going to be going over Principles of Biochemistry Chapter 4, the 3D structure of proteins. Specifically, we'll talk about the secondary, tertiary, and quaternary structures of proteins, as well as protein denaturation and folding, and a few ways to analyze protein structure. First, let's get a brief overview of protein structure. So, proteins can fold in different ways to achieve different conformations. And a protein in its biologically active conformation is a native protein. So a native protein is a protein in its functional folded conformation. Proteins can also have a stability, which is the tendency of a protein to maintain a native conformation. A protein's stability can be influenced by a number of factors, such as the hydrophobic effect. And this is the predominant force that influences the stability of a protein. There's also hydrogen bonding and ion pairs, or salt bridges. And these two are the result of polar group interactions. And then we have van der Waals interactions, which are very weak individually, but can collectively produce a substantial effect on protein stability. So there are 4 different levels of protein structure. The first one is the primary structure, and this refers to the actual sequence of amino acids. Next, we have the secondary structure, and that refers to the local spatial arrangement of the polypeptide backbone. Then we have the tertiary structure, and that refers to the 3D arrangement of all the atoms in a polypeptide chain. And then quaternary structure refers to the 3D arrangement of multiple polypeptide chains. So tertiary structure is still just one polypeptide chain, quaternary is multiple. And not all proteins have a quaternary structure. Some of them stop at the tertiary because they're already biologically active. Let's begin with the primary structure at the peptide bond level. Peptide bonds are formed through a reaction known as dehydration synthesis. And this reaction can also be described as condensation or nucleophilic acyl substitution, or simply just dehydration. Let's go through the mechanism of how the peptide bond is formed. So here we have 2 amino acids, and the alpha carboxylate of one of them is going to attack the protonated alpha amino group of the other, and it's going to steal its proton. Then, the alpha amino group of the other amino acid is going to attack the carbon center of the alpha carboxyl group and form a tetrahedral intermediate. Then, the alkoxide will reform the carbonyl and kick out the hydroxyl group. This hydroxyl group will then deprotonate the nitrogen, and you'll finish forming your peptide bond. So now we have our dipeptide, and we can see the peptide bond formed here between the alpha carboxyl group and the alpha amino group, and this functional group is now called an amide. Now the peptide bond is quite rigid, and that's due to resonance stabilization. So let's go through the resonance structures. So here we have the peptide bond how we usually see it, but the nitrogen can use its lone pairs to form a double bond and force out the electrons from the carbonyl pi bond, and we'll get this structure here that looks pretty structurally similar to an enolate. We can also have these electrons go back to the nitrogen, and we'll get this resonance structure. Now, this last resonance structure we drew is actually the least stable of the three, and the most stable would be the one we started with. So the peptide bond is rigid and planar as a result of partial double bond character. And that means there's no free rotation for the peptide bond. However, there is free rotation around the other bonds in the polypeptide backbone, and these angles that the bonds form are called dihedral angles. So there are three different types of dihedral or torsion angles: phi, psi, and omega. And these dihedral angles are formed as a result of free rotation, with the exception of the omega angle. Phi describes the alpha amino to alpha carbon bond angle. Psi describes the alpha carbon to alpha carbonyl bond angle. And omega describes the alpha carbonyl to alpha amino bond angle. And this one will always be 180 degrees, because as you recall, there's no free rotation about the peptide bond. The other two types of dihedral angles can vary from negative 180 degrees to positive 180 degrees. So here is a generic polypeptide backbone, and here would be the phi bond angle. This would be psi, and over here would be the omega, which is 180. Here's phi, here's psi, here's omega. Now there can be different combinations of phi and psi, and they can be either unfavorable or favorable depending on the situation. They can be unfavorable due to steric hindrance between atoms in the polypeptide backbone and side chains. But they can also be favorable due to the ability to form favorable hydrogen bonding interactions along the peptide backbone. And a way to visualize these combinations is by looking at the Ramachandran plot, which shows the distribution of bi and psi dihedral angles found in a protein. So this is what a Ramachandran plot looks like, and this is taken from figure 4-8 in your textbook. Let's go over how to read one of these plots. So the blue regions over here indicate favorable bi and psi dihedral angles. The darker the blue, the more favorable it is, and therefore the more common that combination is going to be. And these combinations are favorable mainly because they have little to no steric hindrance. So for example, if we were to look at the most favorable phi and psi combinations for the right-handed alpha helix, a type of secondary structure, the most favorable would be if phi was around -150 to -60 degrees, and if psi was around -60 degrees. It is with these values that we get the most favorable darker blue shaded region. And this is for the right-handed alpha helix. You can use the same method to determine the most favorable dihedral angle combinations for the other different helices, as well as the beta sheets. Now that we've had mention of some of these secondary structures, let's talk more in detail about them. So there are two main types of secondary protein structures. The first is the alpha helix, and these are stabilized by hydrogen bonding between nearby residues. The other main type is the beta sheet, which is also stabilized by hydrogen bonding, but between adjacent segments that may not necessarily be nearby. And let's take a closer look at alpha helices first. Like I said before, the alpha helix is held together by hydrogen bonding. The shape is typically a right-handed double helix with 3.6 residues per turn. And this is known as the pitch, or repeated length of the alpha helix. The shape can also be described as a compact spiral rod, and the helical diameter of an alpha helix is about 10 to 12 angstroms long. The peptide bonds in an alpha helix are positioned roughly parallel to the helical axis, while the side chains are positioned roughly perpendicular to the helical axis. Now, as I mentioned before, the typical shape of the alpha helix is a right-handed double helix. What I'm drawing here is a left-handed double helix. In contrast, here is what a right-handed double helix looks like. And it might be kind of hard to tell what's going on in these simple drawings, so there's a more detailed description on figure 4-3 of your textbook, as well as box 4-1, which provides a nice explanation between the differences. But here would be the 3.6 residues, the pitch, and that's about 3.4 angstroms long. We also have to remember that we still have a C and N terminus, and that's going to strengthen the net dipole of the alpha helix. This means you'll more likely find positively charged residues near the carboxyl terminus, and on the other hand, you'll find more negatively charged residues in the other end, toward the N-terminus. Now let's talk about the beta sheets. Just like the alpha helices, these are also held together by H bonding. And beta sheets are when a polypeptide chain is folded such that the strands are connected laterally by hydrogen bonding. And there are two types of beta sheets, parallel and antiparallel. So parallel beta sheets are when you have the two strands lined up parallel to one another, such that they have the same amino to carbonyl orientations. And as a result, the pattern of H bonding occurs in a distorted zigzag fashion, which I'll demonstrate below. So we can have hydrogen bonding over here, here, and here, and then it's also assumed there's also here and here as well. They're just off screen. Another thing to note is that the repeating period for parallel beta sheets is going to be about 6.5 angstroms long. Now let's talk about the antiparallel beta sheets. So the antiparallel beta sheets are lined up antiparallel to one another, such that they have opposite amino to carbonyl orientations. And in contrast to the parallel, for the antiparallel, H bonding occurs in a sort of parallel fashion. It can also be described as being an inline pattern. So we would have hydrogen bonding occur over here, 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 and here. And the repeat period for antiparallel beta sheets is going to be a little bit longer than the parallel at 7 angstroms long. One more thing about antiparallel beta sheets is that they can contain an additional type of structure, which the parallel beta sheets don't have. These structures are called beta turns. And a beta turn is a 180-degree turn that connects the ends of two adjacent segments of an antiparallel beta sheet. And each beta turn involves 4 amino acid residues, which often contain glycine or proline. Now let's move on to the more complex tertiary structures. Tertiary structures result from the folding of alpha helices and beta sheets of the polypeptide chain. These structures are stabilized by a number of interactions, such as hydrogen bonding, ion pairs, or salt bridges, the hydrophobic effect, van der Waals interactions, and disulfide bridges. And these ones may be present in some tertiary structures more than others. For example, collagen versus fibrin, and fibrin is a silk protein. Collagen has lots of disulfide bridges in its structure, which gives it the tough and rigid property that we're familiar with. On the other hand, fibrin has very little of them, allowing silk to be flexible in nature. Then we have quaternary structures. Quaternary structures result from multiple polypeptide chains, that is, multiple tertiary structures coming together. The polypeptide chains, called subunits, are held together by hydrogen bonds, ionic interactions, and hydrophobic interactions. An example of a quaternary structure is hemoglobin, which is composed of 4 polypeptide chains. It has 2 alpha subunits and 2 beta subunits. And the alpha subunits are identical to one another, so they would be protomers, and same for the beta subunits. And again, as mentioned before, not all proteins have a quaternary structure. As we get to higher levels of proteins, we can learn to classify them into 4 general classes. Many higher level proteins can fall into one of the following general classes. First, we have fibrous proteins. These are polypeptide chains that are arranged in long strands or sheets. For example, collagen is arranged as a long strand, and silk fibrin is arranged as a long sheet. We also have globular proteins, which are polypeptide chains that are folded into a spherical or globular shape. These can be things like enzymes and immunoglobulins. We also have membrane proteins, which are polypeptide chains embedded in hydrophobic lipid membranes. And these could be things like membrane receptors or protein channels. And then lastly, we have intrinsically disordered proteins. And these are polypeptide chains that lack stable tertiary structures. And this is due to the fact that these proteins often lack a hydrophobic core. And they are also typically characterized by high densities of charged amino acid residues, such as lysine, arginine, and glutamate. It's also worth noting that all of these classes are structurally distinct from one another. Now let's talk about protein denaturation and folding. Proteins have evolved to operate at particular conditions, such as pH and temperature. When a protein is subjected to extreme changes in these conditions, then protein denaturation may occur. Protein denaturation is when the protein undergoes loss of a 3D structure in the form of partial or complete unfolding. And this loss of structure results in the loss of function for the protein. However, when they are returned to their ideal conditions, denatured proteins can become biologically active again through a process called renaturation. On the topic of protein folding, some proteins can't fold on their own, so they need some help. And this help comes in the form of chaperone proteins. Chaperone proteins can facilitate correct folding pathways or provide microenvironments in which folding can occur. Errors in protein folding can cause or contribute to the development of serious genetic disorders or disease. Some examples are type 2 diabetes, Alzheimer's, Huntington's, and Parkinson's. And lastly, we'll talk about a few methods of analyzing protein structure. There are several ways that you can analyze protein structure. The first that we'll talk about is X-ray crystallography. With X-ray crystallography, you have some kind of X-ray source, a protein crystal, and a detector. The X-ray source is going to fire an X-ray beam at the protein crystal, and that's going to get diffracted into various parts of the detector. The detector records all these diffraction patterns and uploads it into a computer, where a map of electron density can be created. And subsequently, a crystal structure of your protein will be generated. However, the protein crystal can't tell us everything about the protein, so we have other methods too, such as nuclear magnetic resonance, or NMR. NMR is a helpful technique for analyzing the more dynamic characteristics of proteins, such as any conformational changes, protein folding, and intermolecular interactions between the protein and other molecules. Lastly, there's cryo-electron microscopy, or cryo-EM. And this technique involves a sample which contains multiple copies of the structure of interest that is quick frozen in vitreous or non-crystalline ice, and then is observed using an electron microscope. And that's all I have for this video. I hope it was helpful, and thank you for watching.